Hi, I'm Jillian Swinford. I'm a natural resource specialist at Parks and Wildlife. One of the projects I've been working on recently is a really big project looking at American eels. This is something that Parks and Wildlife has been doing for about 6 years now. And our project targets specifically the very young life stages of this fish. We call them glass eels and elvers. And I'll talk a little bit more about that in a little bit. But this project spans all the way up to the Louisiana-Texas border up in Sabine, all the way down through Victoria. So it's a very huge area that we cover. We have different sites on different rivers, and the goal is to catch these early life stages as they swim up river from the Gulf of Mexico, and they're trying to come upriver to spend their life up in fresh water. This is an ongoing project with the University of Houston Clear Lake up in Houston, and then also we're also collaborating with some of our inland folks from Texas Parks and Wildlife too, so it's a really big, very collaborative project. And I personally run 3 of our ramp sites down here, and I'm kind of the chief eDNA person for this project. Eels are a really, really weird fish. They have a really strange life history. So they spawn out in the Sargasso Sea, which is way out in the Atlantic Ocean, and then they hatch as leptocephali, which are these small, translucent larvae, very long, ribbon-like, leaf-like, see-through. And then they drift in to the Atlantic and the Gulf on currents, and from there they metamorphose into glass eels, which look more like an eel. They're very small and completely see-through. And they swim up rivers, entering in from our bays and estuaries, with the goal of, getting up into freshwater, where they're going to spend the rest of their life as adult yellow eels. And so that's usually, if you've caught an eel in Texas, it's probably been an adult yellow eel. And then when they become reproductively mature, when they're able to have babies, they swim back down those rivers, back out in the ocean, and all the way back to the Sargasso Sea, where they reproduce once and then they die. And so they have such a weird, strange life history. It makes it really difficult to study them, which is why, we're targeting all of these different life stages with this study. We use eel ramps to capture the glass eel and elver stages, which elvers are kind of like teenage eels. That's kind of the next step. And we use these ramps because eels like to climb. Because they're moving up into fresh water, they climb these natural barriers like waterfalls, or they'll climb, man-made barriers like culverts or even dams. They're very good at it. And so we use the ramps to kind of trick them into climbing up the ramps. We have that interesting knobby medium that they like to use to climb, and then we also give them a flow to attract them to the ramp. How the ramp works is we use a passive siphon, so we basically just use gravity and the flow from the river that we're sampling on to create this siphon that creates the flow down the ramp that will attract the eels. The eels will swim up, and then they will get sucked down into this bucket that we can then check. And unless the water rise is above the bucket, they won't be able to climb out. So these are very specifically designed fishing gears, but they can be very effective if placed in the right area. Basically what we do is we go out to the site. First we take our eDNA samples. eDNA is basically, it's environmental DNA. So what we're trying to collect is the DNA that's just floating around in the water from when a fish swims by. And as a fish swims by in the water, it sloughs off, cells, slime, waste, feces, that kind of thing, all has DNA in it, and potentially we can use that to figure out if an eel or another kind of fish has been in that area within about a week's time, which is a really cool new thing and works really well for, cryptic species, so animals that are hard to catch with your normal gear. So first we take our eDNA samples, and you want to take them first so you don't have any risk of outside contamination, or if you're walking around on the sediment, you might stir up some of that sediment, and we don't want that getting into the sample. So we take those first, and they're just a simple water grab, and we process them all entirely back in the lab. And then from there, we take water parameters, water quality, so that includes like temperature, salinity, dissolved oxygen, pH, and then turbidity, which is basically like how cloudy or clear the water is, more or less. And then from there, we check the ramp, make sure it's flowing well, and then we check our bucket to see what we have caught. And then we'll classify it by species. So we'll measure out all of the fish and crustaceans we catch. So we catch other stuff along with the eels as well. And then we close it up and make sure that there's no maintenance that needs to be done. And then we head out to the next site. So in order to process the eDNA, for us to be able to tell whether there's eel DNA in our sample or not, we take our samples back to the lab and we filter the water immediately. And the water goes through these very fine filters that traps the sediment and the DNA that sticks to the sediment. And from there, we take the filters and we shred them up in a special vial that allows us to, break down what's on that filter. And then from there, we can extract the DNA from that sample, and we can then use it to determine whether we have eel DNA in that sample or not.